Okay, so while you're opening our slides, I'm just going to introduce you. So good morning, everybody. We have a very special grand rounds this morning. We have Dr. Michael Kruer from the Pediatric Movement Disorder Specialist. He's also a human geneticist. He serves as an associate professor of child health, genetics, neurology, and cellular molecular medicine at the University of Arizona College of Medicine in Phoenix. He also serves as director of the Pediatric Movement Disorder Program at the Barrow Neurological Institute at Phoenix Children's Hospital. Dr. Kruer's lab is dedicated to understanding how genetic mutations lead to both common, like cerebral palsy, and rare movement disorders in children, and how genetics can serve as a springboard to improvements in both diagnosis and treatment. Work in his lab is highly multidisciplinary and connects genomics, phenomics, stakeholder engagement, and molecular and cellular neuroscience. His research has been supported by numerous public and private foundations, philanthropy, industry collaborations, and federal grants. And his work has appeared in highly respected publications, including the American Journal of Human Genetics, and also of Neurology, Brain, Pediatrics, Neurology, and Nature Genetics. The Kruer Lab is currently leading an NIH-funded genomics initiative within Cerebral Palsy Research Network, which is a nationwide collaborative of cerebral palsy centers of excellence that leverages a self-contained electronic eye consent platform to facilitate network-wide data sharing for genetic discovering in cerebral palsy, which as pediatric neurologists, we're definitely all very excited about. So with great pleasure, I'd like to introduce Dr. Michael Kruer.
Thank you so much for that kind introduction, and thanks to all of you for the opportunity to speak with you this morning. As Theodore mentioned, I'm going to be talking about some of the recent work in the field, some of which is coming out of our group, some of which has come out of other groups in regard to genetic causes of cerebral palsy. And so here are my disclosures, but none of them are directly relevant to the talk today. So I wanted to start things off with a case. And so this is a 7-year-old boy with global developmental delay that presents to your office for first-time consultation. This patient has spastic dystonic quadriplegia. He experiences recurrent focal motor seizures with secondary generalization despite AED treatment. He's not able to walk independently, and his full-scale IQ is measured at 54. An MRI discloses a malformation of cortical development by hemispheric pachygyria, and whole exome sequencing is done, and it discloses a pathogenic de novo mutation in the TUBA1A gene. And so just for the sake of discussion, I would ask all of you, is he rightfully considered to have intellectual disability? Many people would say yes, particularly based on the neuropsychological assessment and the full-scale IQ. Another question would also be, does he have a malformation of cortical development? And according to current consensus definitions, most would say yes. Is he rightfully considered to have epilepsy? Well, he does have recurrent unprovoked seizures, and so by the ILAE definition, yes, he does. Does he have a Mendelian disease? A single gene disorder. And again, with the identified mutation in TUBA1A, most would say yes. But then I guess the question comes up, does he have cerebral palsy? And here's where we as child neurologists will often diverge. And so one of my favorite parables or stories, if you will, and many of you have undoubtedly heard this, is that of the blind men and the elephant. And essentially, this story goes like this. There's a group of blind men that are walking in the jungle when they come upon an elephant. And each of them reaches out and interacts with a different part of the elephant. So the blind man who reaches the tail thinks that it's a rope. The one who grabs one of the elephant's great legs thinks that it's a tree trunk. But interestingly, I find that a lot of times doing the genetics work that we do, there are some nuances in the language that we use. And so, for example, if you were to consider a single patient that has, let's say, dystonia, sometimes when talking with other clinicians, whether they're neurologists or in other fields, other terms will be used. And so things like dyskinesia, motor delay may be applicable, hypertonia as a form of kind of undifferentiated increased muscle tone. Psychomotor retardation is a term that's still in use. And then I think whether the term CP applies is also commonly asked. And so I think that there's a growing sense that dialogue and words are important. And so for this morning's talk, I'm going to focus on a neurodevelopmental perspective, really looking at the different domains of development. I think that when you're considering the core domains of development, you can think about disorders of cognitive development as essentially representing learning disorders or on the spectrum of intellectual disability. Autism is thought of to impact the language and social emotional domains of development. And similarly, cerebral palsy is the most common cause of physical disability in children. And so we're going to be looking at CP through a bit of a neurodevelopmental perspective. So when I start talking about genetics and CP, there's a number of questions that commonly come up. One of them is well, what do you mean a genetic basis for CP? And the concept is that, well, CP implies a birth injury of some kind, right? And I think that CP as a diagnosis is sometimes used in that way. Although, interestingly, if you actually look to the consensus definition of cerebral palsy, which was established in 2006, published in 2007 by this international group of experts, which included child neurologists among them. Interestingly, the definition of CP is actually agnostic as to etiology. So cerebral palsy, similar to the other developmental disorders, is a clinical diagnosis at its core. And so one cause or another, at least according to the current consensus definition, doesn't necessarily impact the diagnosis of cerebral palsy. Another common comment that comes up is, well, CP is a wastebasket diagnosis anyway. And this is certainly something I heard quite often during my training. And Terry Sanger in the movement disorders field took this head on in this editorial in the Journal of Child Neurology now more than 10 years ago. And this was shortly after the consensus definition was published. But in this article, Terry really considered the different aspects of a CP diagnosis and concluded that if rigorously applied, cerebral palsy actually was a valid construct and a useful diagnosis for patients and also for clinicians. Now, with that said, I'll be the first to say that I think that sometimes the label of CP is used indiscriminately. And so during my training when I was doing botulinum toxin injection training, I had an experience where I was in a clinic outside of my home base, and I asked about the patients that we were seeing that clinic, and I was told that all the patients had CP. And I said, okay. Well, I looked a little bit further at the charts out of interest, and I found out that the patients actually had some potentially important differences. One of the patients had a hypoxic ischemic injury at the time of birth that had resulted in their motor impairment. Another patient had pachygyria. Yet another one of the patients had a traumatic brain injury that occurred during adolescence. And finally, one of the patients had a progressive dystonia. And so I think that this really brought to my attention that sometimes the diagnosis of CP can be indiscriminately applied. And so I think that this really then leads to the question of, well, how do you define CP? And so going back to the consensus definition, cerebral palsy is fundamentally described this way. CP describes a group of permanent disorders of the development of movement and posture that causes activity limitation, so it has a functional impact, and it's attributed to a non-progressive disturbance that occurred in the developing fetal or infant brain. And so fundamentally, CP is considered a developmental disorder. And so it was this perspective that led myself and other members of the International CP Genomics Consortium and other members of the pediatric movement disorders community to put forward this opinion piece, where we suggested that the identification of a genetic etiology shouldn't necessarily lead to the undiagnosis of cerebral palsy. That CP perhaps represents an etiology, whereas, the genetic finding actually represents an etiology, but CP may represent a valid clinical diagnosis in and of itself. And so I think that this, leads to more questions. And one of the follow-up questions that often comes about is, well, how can we call all of these the same thing if the etiologies are so different? And people might point to the differences, for example, in hypoxic ischemic injury and something like pachygyria. However, I think it's interesting to consider that both of these etiologies can cause cognitive problems that we might diagnose as intellectual disability. Another comment that comes up is that CP is so variable. But I think it's, again, interesting to consider the parallels. And if we think of the epilepsies, Ohtahara syndrome is quite different from Orlandic epilepsy in many ways, although we both, I think, rightfully consider those to be forms of epilepsy. So this really speaks to the importance of considering diagnosis versus etiology as we're starting to talk about these concepts. One of the comments that sometimes comes up is, well, I guess everything is just CP then, right? And I disagree. I think that this is something that we try to emphasize to our trainees. And if you go down to the definition of CP on the bottom right, you can see that this actually does provide some degree of clarity. And so, for example, I would not consider juvenile Parkinsonism to represent a form of CP because it involves an inconsistent movement disorder. Paroxysmal dyskinesia is a non-permanent condition, and so typically is not considered to be a form of CP. Duchenne muscular dystrophy, fundamentally neuromuscular, not a movement disorder, and so does not represent CP. Something like PCAN, pantothenate kinase associated neurodegeneration, is fundamentally a progressive disorder, and so would not be considered to represent cerebral palsy. Similarly, a school-age onset dystonia does not occur in the developing brain, and so would not rightfully be considered CP. And finally, spinal cord injury is not a brain disorder, and so would not be considered to represent a form of CP. And so this is unpublished data, just a survey of our practice at Phoenix Children's Hospital in the movement disorders section, but we find that although we see CP quite frequently, tic disorders actually represent the majority of cases that we see, but a sizable proportion of our practice are actually these rare pediatric movement disorders. And so I think that there are some important similarities, but also some important differences between these categories of disorder. So you might be saying at this point in time, well, isn't this all just semantics? Does this really matter? And I think that I would argue that it does, that words matter. And give the example, sometimes I'm on the wards and I hear the terms like this or comments like this. This is a 23-year-old male with developmental delay. And the question is that really the most appropriate diagnosis at this point in time? And so I think it's important to recognize that families really do hinge on what we do and don't say. And again, I would argue that words do matter. This was a very nice article here in the bottom right, written by Michelle Schusterman, who's a mom of a daughter that was originally diagnosed with cerebral palsy. And in this article, Michelle writes about how that diagnosis really led her to have certain expectations. But she describes how, in retrospect, she wasn't prepared for the recurrent seizures that her daughter experienced, for the cognitive problems that she had, or the cortical visual impairment that affected her daughter. And so, again, I would argue that these developmental diagnoses do in fact matter. because they help to set a common language both between specialists, but also for our patients and their families. So hopefully I've given you some food for thought, but I do think that there is perhaps a need to continue for all of us to think integratively. And I think there's a increasing recognition that many of the common developmental disorders that we see are actually collections of individually rare conditions. And particularly with the explosion in genetic etiologies being detected, this is probably more true than ever in child neurology practice. So when I first started my laboratory and our research program, we were working on single gene disorders, and I got some advice at the time that I should shift my focus because single gene disorders, monogenic conditions, or sometimes called Mendelian disorders, were essentially solved and that there wasn't going to be much more to do. But this is a very nice article that was published by several leaders of the Centers for Mendelian Genomics just now a couple years ago. And I think what this shows in the top left here is that if you look at the recent years, since the advent of exome sequencing, we've seen a steady rise in the number of newly discovered single gene disorders. And these genes have been known to exist through the Human Genome Project, but their association with human disease is what's actually still being discovered very actively. If you turn to the figure here on the right, you can see that if you look at the approximately 20,000 different human genes that exist in our genome, we actually know of a minority of them. And so what this figure tells us is that at this point in time, of course, time will tell if every human gene is ultimately associated with the disease. But it looks like that we're still in a period of very active disease gene discovery, and still probably have quite a bit to learn about human genetics. All right, so hopefully that was some helpful background, but at this point, I'm going to shift a little bit and talk more specifically about some of the recent findings in cerebral palsy genetics. And I think a lot of times when we talk about cerebral palsy genetics, one important concept, I guess, to discuss right up front is that most of the recent work has actually focused on Mendelian conditions, single gene disorders. And so where the early work was essentially focused on candidate genes that might increase an individual's susceptibility to an acquired insult, something like a hypoxic ischemic injury, for example, or like a premature birth. Some of the more recent work, including our own work, has actually started to look at changes in single genes of high effect size. So these are changes in single genes that essentially lead to mutations and are thought to tip the scales in between health and disease or a developmental disorder in and of themselves. And so when talking about these genetic changes of large effect size, we're talking essentially about copy number variants at the genomic level, as well as single gene etiologies. However, even though recent work has focused in this area, I think it's important to recognize that there's undoubtedly more to the story. And particularly in the cerebral palsy space, there are some very interesting questions about gene by environment interactions that I think we'll be able to ask. So over the last 10 years or so, there actually has been slowly accumulating evidence that genetics and genetic mutations may be important for cerebral palsy. And you can see here a representation of some of the important initial studies that particularly identified copy number variants in individuals with cerebral palsy. Some of this work focused on so-called cryptogenic CP, which were individuals that had CP of unknown etiology. So typically kids that had a clinical diagnosis of CP, but didn't have the typical risk factors. Interestingly, other groups took a different approach. And so the Toronto group, for example, specifically looked at individuals with hemiplegic CP, which many, including myself, thought would be relatively under represented in terms of genetic causes. And quite importantly, they showed that even in the hemiplegic form of CP, there were increased numbers of copy number variations at the genomic level. Finally, the Adelaide, Australia group put out this important paper published in Molecular Psychiatry that showed that there was likely to be important single nucleotide variants related to CP. However, much of this early work, unfortunately, had limitations because of the small cohorts that were involved and the inability to do statistical and functional validations of these findings. And so we actually formed an international consortium, and I was very, very pleased to help lead this effort. And this was published just a little over a year ago in Nature Genetics. But this was the result of really a worldwide collaboration where we put together our collective data and doing some advanced statistical and functional genomic analyses. We were able to show for really the first time firm evidence that a substantial proportion of CP cases are in fact due to genetic mutations. And so this work really establishes genetic causes as an important contributor to CP, in addition to long accepted risk factors like prematurity, hypoxia, ischemia, and pre and perinatal stroke. And so just in the short time since then, there have actually been some outstanding follow-up papers that have been published. The one on the top left, led by our collaborator, Michael Zeck, looked at dystonia genes and found, interestingly, that dystonia genes as a whole, even those with adult onset, were enriched for genes that had previously been associated with neurodevelopmental disorders. The Munich group also found evidence for dystonic forms of CP to have a strong genetic influence. The paper on the bottom left showed that there was a substantial contribution of genetic variants to cerebral palsy in a clinical population, and suggested that there was somewhere between 10 and 30% of individuals with CP, depending upon how they were ascertained, that might actually have a genetic etiology for their symptoms. More recently, this paper in Annals of Clinical and Translational Neurology, led by Maya Chopra and Sid Srivastava at Boston Children's, really showed very nicely that genetic causes are an important contribution to clinical cerebral palsy, and also made the case that individuals with CP, even if they had known risk factors, could sometimes be found to have demonstrable genetic mutations. Similarly, this paper, led by our colleagues Helly May and Jason Carmel at Columbia, also demonstrated an important role for genetic mutations in cerebral palsy of diverse etiologies. And so I think that the field is rapidly growing. However, with that said, there have already been some interesting surprises along the way. This was something that to me was quite interesting. So these are just two representative MRI images that were included these are two patients that were in our Nature Genetics cohort. And so both of these individuals were seen at very good centers. They had MRIs read by outstanding neuroradiologists. One was read as subcortical and periventricular T2 hyperintensities that are suggestive of an in utero ischemic event. The other was read as the T2 or flare hyperintensities, likely secondary to gliosis from a prior hypoxic ischemic insult. What was interesting, though, was that neither of these individuals were known to have had any complications with their pregnancy or the delivery. Both moms had received the routine prenatal care and had actually done quite well. And so when we did exome sequencing in these patients, interestingly, we found what were compelling genetic changes in each of these genes. And so it was thought that mutations in these respective genes actually accounted for the MRI appearance rather than some kind of occult hypoxia ischemia. Another thing that we recognized with the Nature Genetics work was that there's substantial clinical and genetic overlap among the major neurodevelopmental disorders. And clinically, we've known for some time, clinically and epidemiologically, that there's an overlap between CP and other NDDs. So for example, more than half of individuals with CP are typically thought to have some degree of intellectual disability. More than 40% are thought to have epilepsy, and autism spectrum disorder is thought to co-occur in about 6 to 10% of cases of CP. And interestingly, we found that many of the genes that we identified in individuals within our cohort with cerebral palsy had actually previously been implicated in these other forms of NDD. And we think that there are both some things that are not surprising and potentially surprising that are embedded in this data, but it's going to take higher numbers to sort it out. But our preliminary findings suggest that a couple things are going on. One, that there is certainly overlap among these major neurodevelopmental disorders. And so we ascertained our patients based on whether they had cerebral palsy or not. Whereas investigators that study epilepsy typically ascertain their cases based on whether they have epilepsy or not. In many cases, these individuals will have both of these conditions. And so that's to some extent what we're looking at here. However, there's also examples where genes were known to represent epilepsy genes, for example, and we have seen mutations in individuals with cerebral palsy that also seem to be due to mutations in the same gene. So this is suggesting that in some cases, mutations in key genes that govern neurodevelopment might have variable effects and can actually lead to different clinical phenotypes. Interestingly, when we looked at the genes that we identified as a whole, we found that these genes weren't just randomly distributed throughout the genome, but actually represented important players in early brain development. And so what you see depicted here on the upper left is a couple of induced pluripotent stem cell neurons that my former postdoc, Brandon Guida, developed. One is from a patient carrying a mutation in row B. The other is from a control. And you can see that there are some substantial differences just qualitatively in terms of neurite outgrowth. One thing that was quite interesting was that we found that cytoskeletal pathways were highly impacted by the mutations that we found in our cohort. And this was intriguing to us because of what we know about the role of the cytoskeleton in normal brain development. And so this is a video that really shows, this is a movement disorder neurologist's worst nightmare. Unfortunately, my video is not playing, but essentially what you would see is you would see that coming from the soma of this neuron in culture, there are actually waves of actin that will come and propagate along the structure of the developing neurites. And those waves of actin seem to be crucial in forming the normal morphology of developing brain cells. Essentially, we think that disruption of this normal process may lead to some of the abnormalities of connectivity that we see in some forms of CP. And so just since the publication of that initial cohort, we've started to see more and more single gene causes of CP beginning to emerge. And just in the last year or so, this is just a brief example of the different papers that are starting to come out. And so I think that it's actually quite an exciting time for cerebral palsy associated gene discovery. Now, we've also come to find some very interesting parallel observations as we've begun to study these processes. And so this is work that my postdoc Sarah Lewis has done using Drosophila, the fruit fly model. But we actually know precious little about the molecular and cellular basis of dystonia. And so Sarah has started to dig into this with studies in the fruit fly, beginning with studies in fly models of single gene forms of CP. And so we've known that dystonia seems to represent an important movement disorder, particularly an important subtype of cerebral palsy. We also know that dystonia seems to be related to abnormalities of neuronal circuits. But again, those mechanisms have proved elusive. But to summarize a great deal of work very quickly, Sarah has started to focus on the EIF2 alpha pathway, which is a key pathway involved in cytotoxic stress responses. And this gene actually governs normal translation of proteins during both development and during times of stress. And what Sarah's work has started to really highlight is that, interestingly, it seems like there's a sweet spot for EIF2 alpha signaling, and that too much or too little signaling through this pathway actually impacts neuronal development in the animal model, but also will impair locomotion in the fly. So put another way, this affects the fly's ability to walk. What's been quite interesting is that Sarah has gone on to show that these impairments in normal walking behavior in the flies to some extent seem to be due to dystonia-like movements that are elicited in the fruit fly. And although I don't have time to go into all the details, the work has also suggested that if you genetically manipulate this pathway and either turn it up or turn it down, that it will fundamentally alter synaptic connectivity and morphology at the synapse. And so we're very excited to see where this will lead us. The question that finally comes up in discussing this work is do these patients really have CP? And this is a screenshot taken from OMIM, Online Mendelian Inheritance in Man, which is looking at a particular neurodevelopmental gene, the GRIN1 gene, which is a subunit of the glutamatergic NMDA receptor. As you can see, and as many of you are undoubtedly familiar with, there's a trend in genetics to name the disorders on a single gene basis. And I think that this represents a valid construct. So for example, in genetics, this might be referred to as NDH-MSD disorder. But I think what's starting to be argued in the field is that these individuals may have this very specific genetic diagnosis, but they also have legitimate developmental diagnoses. And so many of these individuals will have seizures and in fact have an epileptic encephalopathy. It's being increasingly argued that these individuals can rightfully be considered to have both epilepsy and CP, and this rare single gene disorder, that there's value in each of these connections. So some of you might be asking, all right, well, CP can be genetic, so what? What does that really change? And again, this is a work that's done by Sarah in my lab, but she's become very interested in this question and started to look at it across cohorts in different published data sets. And what she started to find, quite interestingly, was that whether you look at things from research cohorts or clinical cohorts, if you apply the same criteria, and these are modified forms of a ClinGen rubric, these genes are starting to have potential implications for clinical management right now. And so this means that a genetic finding at the current time can actually impact what we do for these patients, potentially preventing complications, or in other cases, potentially offering new options for treatment, which I think is particularly exciting in the CP field, since new treatments have been hard to come by for some time. And one thing that's important to mention is although the level of actionable genes is somewhat surprisingly high, there is quite an evidence gap. And so in general, the quality of the evidence supporting the potential actionability of these genes is quite low. And so I think it's very safe to say that additional studies are certainly needed. I'm going to skip this in the interest of time, but I think that in terms of thinking about implications of some of the findings for cerebral palsy, the genetic findings can actually help us to sometimes reach for the best treatments first. And so a nice example of this is with the beta-catenin gene, CTNNB1. This gene so far actually represents the most common single gene cause of cerebral palsy. And individuals with mutations in beta-catenin do have variable phenotypes, but based upon the developmental neurobiology associated with beta-catenin, the Toronto group actually hypothesized that dopaminergic neuron development could be particularly effective. And so in this article published in Movement Disorders, they actually described their experience using levodopa for an individual with beta-catenin mutation, and found that there was a demonstrable response. Now, this was not a night and day response, but this was an example of a situation in which perhaps treatment could actually be directed based upon genetic findings, so that we're not simply relegated to trial and error. Another aspect of this that's quite interesting for us involves this scenario. And so this is a young patient of mine with a dyskinetic form of CP, and he has quite substantial chorea. And of course, my codecs are not going to work again, but what you would see is that for this young man, he has choreic intrusions throughout the gait cycle that affect his ability to just walk down the hallway. Remarkably, he's still able to walk despite this, but he was found to have a mutation in a gene called ADCY5. And interestingly, our colleague Emmanuel Rose reported just recently that individuals with mutations in ADCY5, which is an adenylyl cyclase gene, may actually be caffeine responsive. And so this initial report described a single family with a marked caffeine response. And this was, of course, picked up by news outlets who advertised this as a potential new treatment for CP. Now, I think that it's important to recognize that this does not necessarily mean that caffeine will be of use for all individuals with CP, but it is an interesting observation that suggests that we might be able to personalize some of our treatments based upon some of the genetic etiologies that we're starting to detect. All right, well, I'm going to start to wrap up here, but I did want to tell you one last story. And this is related to what our studies of cerebral palsy genetics are not only telling us about cerebral palsy, but what they're starting to tell us about normal human brain development. And so this story actually involves a situation where we have human genomic knockouts of PGBD5. And so this is a ultra rare disorder. We only know of 2 families and 4 patients so far. And this is so far unpublished work. But interestingly, both of these families, one is Turkish, one is North African, each had 2 children with a mixed neurodevelopmental disorder that you can see in the pedigrees here. Now, what was quite interesting was that both of these families actually came from regions where consanguinity is quite common. And what we do know is that in areas where interrelation of blood relatives occurs, that sometimes we can see for a brief period of time human genomic knockouts, meaning severe mutations that affect both copies of a gene and lead to complete loss of that gene's function. And this is precisely what occurred in this situation, in which we had one premature stop mutation in one of the families and a premature frame shift mutation in the other families that both led to complete loss of this PGBD5 gene. Now, these children were found to have severe intellectual disability, to have an epileptic encephalopathy, and to have mixed spastic dystonic cerebral palsy. Now, what was quite interesting about this particular scenario, this particular gene, is based upon the gene's function. Because just recently, our collaborator, Alex Kensis, and his group at Sloan Memorial Kettering were able to show that PGBD5 actually represents a bona fide human cut and paste transposase. And so this is an example of a so-called jumping gene. And PGBD5 expression is known to affect essentially loci on every chromosome within the genome and lead to genomic instability. So Alex and his group originally became interested in a potential role for PGBD5 in neoplastic transformation and the development of human cancers. Now, interestingly, we know that the human brain is actually a genomic mosaic. And within recent years, there's been some fascinating work that tells us that mutations that are potentially undetectable in the blood or even the saliva can nevertheless profoundly affect brain development and function. And so, for example, there have been some very nice papers that have shown that copy number variations in the genome can actually affect autism risk by affecting neurons within the genome. We also know that, for example, cortical dysplasias have a high incidence of local genomic mosaicism, where clones of developing neurons, for example, will actually be fundamentally different than the surrounding tissue. We also know that there's quite a landscape of somatic mutation, and this is seen both in neurotypical individuals, but also in individuals with disease. Although so far, the work in this area has really focused on neurodevelopmental disorders. So this brought up a very interesting question for us, which is, if you think about PGBD5 and its function, why would a gene that causes genomic instability be essential for normal brain development? So if you remember, I told you that PGBD5 is essentially serving as a jumping gene and leading to changes in the genome. Why would the loss of this function actually be thought to be associated with such a severe neurodevelopmental disorder? Well, we're still in the midst of this work, but I think it's important or potentially relevant to think about early brain development, in which we know, based upon decades of work in both human postmortem samples as well as mouse studies, that millions of neurons will actually commit suicide. They'll go through programmed apoptosis during crucial phases of normal mouse and human brain development. And it's thought that this actually occurs at several different points during development, but that this is a crucial part of the normal development of the cortex. It's thought, for example, that controlling the fate of these precursors actually can control brain growth to some extent. And it's also thought that there is a potential connection with activity-dependent plasticity, so that to some extent, during early brain development, it's only the neuronal connections, the circuits, that are most relevant to the organism that are actually retained. And those neurons that are part of circuits that are not utilized or not needed will undergo programmed cell death. Now, interestingly, when you look at the expression of PGBD5 in both the human and the mouse, it's overwhelmingly expressed in brain. And so, as I mentioned, this is still unpublished data, but we were able to take the clinical MRIs from our patients, and with the help of our colleague, Alex Pagnosi, in Australia, we were able to do volumetric analyses and show that these patients actually had severe abnormalities in terms of their cerebellar volumes in particular. Now, this was quite interesting because if you jump ahead to the mouse data, we actually were finding that the mouse brain morphology mirrored this at certain points in development. So the mice were found to have both motor and cognitive deficits that I don't have time to tell you about in detail, but suffice it to say that the knockout mouse actually seemed to recapitulate key features of the human neurodevelopmental disorder. Now, we think that this might actually be connected and that the ultimate etiology of PGBD5 related disorder might relate to normal apoptosis within the developing brain. So this is genomic data, whole genome sequencing data that's showing us that there are abnormalities within the genome of the knockout animals compared to the controls. And essentially what we're finding is that these abnormalities of the genomic deletion do seem to correlate with apoptosis. And so we think what ultimately might be happening here is that this might be a case of brain microevolution. So we think that PGBD5 might contribute in the normal context to genomic diversity within the human brain, leading each neuron to be different than its neighbor to some extent. And it's this genomic diversity that's thought to potentially play an important role during normal human brain development. The loss of this diversity, intriguingly, seems to lead to a severe mixed neurodevelopmental disorder. So obviously we have a lot of gaps to fill in, and this work is still underway, but this is starting to lead us in very, very interesting directions. I'm going to close just telling you a little bit about some of our current studies. So as Theodore mentioned in the very beginning, we've partnered with the Cerebral Palsy Research Network. We serve as a contributing site to the CPRN, which is a national collaborative of CP centers. And one of the beauties of this network is that it utilizes the electronic medical record. So as we as clinicians see patients during the course of our clinics, we're entering data just to complete our clinical documentation. But the CPRN structure allows data to be extracted from our notes and actually flow to the Data Coordinating Center at the University of Utah. The net result of this is that the CPRN clinical registry currently contains clinical data for more than 5,000 individuals with CP, and that number is expected to double at least within the next several years. We've partnered up with the CPRN to actually conduct genomic studies on CPRN participants. And so using privacy-preserving records linkage through a software called Datavant, we've been able to securely connect this clinical data housed at the Data Coordinating Center with the whole exome and whole genome sequences that we're developing for the participants, which is proving to be a very, very rich data set. We're coupling this with what we're calling the I-consent. So particularly with COVID, this totally changed how we were approaching the consent and enrollment of participants. We've shifted to an electronic I-consent, and in more recent weeks have been coupling this still with in-person consents, but this has allowed us to really extend our reach much further and make the study accessible to far more families than we could using traditional enrollment alone. And so essentially the I-consent serves the dual purpose of providing information about the study through a series of web pages and short videos, question and answer formats. And then if families like what they see and they're interested in participating, they can sign up directly within the study app itself. In closing, I just wanted to mention this work, which was led by our colleague Buma Aravamutan at Wash U, and it involved several of us within the Cerebral Palsy SIG within the Child Neurology Society. And essentially this paper published in Pediatrics within the last year showed that even so-called cerebral palsy experts are variable in their diagnosis of cerebral palsy. And so I think that this paper represents a bit of a call to action. But what were the drivers of some of that variability included hypotonic forms of CP, as well as forms of CP that included a genetic etiology. And so I think that this is really a call to action, as I said, and I think it's important to keep talking. Fundamentally, I'm a big believer that as much as possible, we need a common language between providers and across specialties, and that this is critical for our patients and their families, so that they're not hearing different things from different people. So with that, I'd be happy to take any questions you might have, but thank you so much for the chance to talk with all of you and to share some of our recent findings.
Thank you so much for such a wonderful talk. If anyone wants to unmute, they can go ahead and unmute for any questions.
Thank you so much, Dr. Kruer. It was an amazing lecture, and I think we all learned a lot. I just wanted to ask about in one of your slides, you mentioned genetic mutation, like a risk factor versus etiology. So in that aspect, if you want to say like a baby that has, like, let's say HIE, if we can detect some genetic mutation, should we attribute the mutation as a risk factor for that baby to develop CP, or the genetic mutation to be the cause for actually the initial insult, like the mutation was the culprit in like baby to have an HIE in some sort?
What a fantastic question. I don't think I have the answer to that. I think that, but you bring up a very important issue. And this is something that we and others are just now starting to dig into with our genomic data and with other methods. But as I mentioned, a lot of the initial work focused on kind of the low-hanging fruit, you know, the individuals that had a clinical diagnosis of CP and had no other obvious compelling reason to have CP. As we're starting to expand the reach of sequencing, we're starting to encounter a scenario like you just mentioned more and more often, where individuals seem to have legitimate hypoxic ischemic insult, for example. They have a difficult start to life, and yet we also identify what seems to be a bona fide genetic mutation. And I think that the answer is probably going to be that there's an interaction, that there's an important gene by environment interaction. And I think at the individual patient level, that's something that's kind of within the realm of the art of medicine right now, to try to figure out what those different factors are. But I think from a research standpoint, that's something that we're hoping to have more data on here in the near future. Because I completely agree, there's this sense in the field that those kids that do have single gene disorders and do have genetic mutations, they're probably predisposed to those more difficult starts to life. But I think we'll have to see where the data takes us.
Thank you.
Good morning. One more question. Thank you again. Your grand rounds reminded me of what we used to, ascribe as double hit theory. And saying that, yes, there's HIE. And again, we see so many premature babies, but, not everybody comes down with CP. So what's different about them? Was there something else that predisposed them? And so I think, our interest in defining CP, I'm very happy, because this has been a nightmare for all pediatric neurologists, purists, who want to say, when do we call it static? When do we call it progressive? And so on. That we are, thanks to genetics, we're looking more into it. And your work has highlighted for all of us that we need to really focus on this. And I thank you again.
My pleasure. Thank you so much. it looks like there was a question in the chat. This essentially said, you showed a brain MRI that had been read as a perinatal stroke and suggested it was misread and that the CP was due to a genetic cause. Do you think this is common? How did neuroradiologists feel about it? How do you prove it? Yeah, this kind of builds upon what we were just discussing. Is there some, is there legitimate hypoxia ischemia that's occurring in some of these cases? Is it associated with the genetic mutation? Or in fact, could this be a quote unquote misread? I think that we want to be careful about how we approach this, and just as much as possible try to let the data lead us. Interestingly, Michael Chevelle and his group in Montreal showed a number of years ago that when you look at so-called PVL, that PVL often seems to be a different animal in children that were born preterm compared to those kids that have PVL identified on term MRIs. And they suggested that there might be different contributing factors, there might be different etiologies, different risk factors. And I think to some extent our findings are really starting to indicate this. I guess at this point in time, I would just urge for us all to just put on a kind of a critical mindset, as we always do, and try to think about, for example, with the diagnosis of CP, to always think about what the potential etiology might be. And similarly, when we're faced with a MRI appearance, to always think about what the etiology might be for that. All right, well, I think we're just about out of time. I have to run the clinic myself, but I hope that all of you have a wonderful rest of your day. Thank you so much for the chance to speak with you, and have a great rest of your day.
Thank you so much for such a wonderful lecture, Dr. Kruer. Take care.
My pleasure, guys. Bye-bye.